Josh, I reflect back the first time we spoke about this, and one of the topics we raised was patient tolerability of ADCs and the fact that actually it's not that great. The patient reported outcomes in this space are, to be honest, pretty poor. Patients don't often like taking these ADCs, although they know they're getting therapeutic benefit. The tolerability, the side effects are not much better than the isolated cytotoxic. It feels like specificity and tolerability and extending that therapeutic window is still a massive challenge in the ADC space, even though that's what these molecules were designed to combat.
Yeah, absolutely. And I think what you're seeing at the moment within the field is that payloads are being thought about, but not as broad as what we would think about them within a small molecule aspect. We go back a couple of years, PDB dimers were the big new thing. It was a really cytotoxic molecule. It went great. It was a DAR2. And in fact, what we've seen is the industry completely turned on its head. We're going for less potent, higher DAR molecules like the topoisomerase. And we're building in that selectivity by the lower potencies and getting that therapeutic window increased from lower potencies, but we're still not cytotoxic.
Mm.
And so what we really want to do is build in another level of selectivity. And where can we do that? From the small molecule world. And we can build in, even if we're not using payloads that are already developed in the small molecule world, taking that thought process around increasing the therapeutic window through PK and really modifying the molecules to ensure that we get that gap.
I think this is a really interesting way of thinking about an ADC, again, as a delivery vector rather than necessarily a way of just delivering a cytotoxic agent. I love the idea of stretching that therapeutic window by building in selectivity through both the delivery and then targeting to the tumor cell. It feels like this is an opportunity beyond just oncology. If you think about most of the metabolic targets we've looked at over the past 20, 30 years, because they are involved in normal cellular metabolism, they have side effects. They have challenges in terms of how we use those therapies in patients. And I wonder whether things like the metabolic space is going to be one of the next big areas for ADC development.
I think so, yeah. I think with the buzz around metabolism anyway, or metabolic therapies at the moment with the GLP-1s, I think it's a big one. I mean, we still have to inject GLP-1s as well.
Yeah.
So the main caveat to an ADC is that we can't go via an oral route when we compare it to small molecules. In fact, we're already in that space with the GLP-1s and people are quite happy to inject themselves at home now. So why not increase the selectivity, increase that therapeutic window, arguably maybe from a more expensive perspective. But as we know, once things become more and more developed, things get cheaper as well. So absolutely the space within the metabolic arena that we can really look at ADCs as another concept.
So how do you see us extending that toxicity window, that therapeutic window, reducing toxicity? I mean, is it just the payload? Is it just the linker? Increased antibody specificity? How do you see that playing out over the next 2 to 5 years?
I think we're probably there with the antibody space at the moment. I like to think of it, I think there's two ways in which you can look at it. And you can either think of it as a large molecule that we're introducing another mechanism of action with the cytotoxin, or it's a small molecule where we're getting a delivery system involved. And I quite like the latter more than the former, because if we strip out that antibody to have just one functionality, and that's delivery, that's the thing that's missing from the small molecules. That's the one that we want to build out as much as possible. So to summarize, really, we're looking at developing the small molecule or the payload in a small molecule asset to be able to actually conjugate it to an antibody and increase the therapeutic window that way. We're creating this selectivity squared sort of aspect where we're building the selectivity into the antibody and we're building the selectivity into the small molecule. And then the linker, I mean, we mentioned it in the last video that the linker's also got a mechanism of action as well. So increasing that selectivity really gets us to that position.
I love that concept of selectivity squared. It almost feels like a bispecific, but beyond a bispecific. So taking that concept of a bispecific antibody, but rather than having two halves of an antibody, which are specific to two separate targets, having the antibody and then the small molecule therapeutic as the second partner of that bispecific modality.
Yeah, I agree. I mean, it's really something that, to me, we're trying to build the most selective molecule. And so we can look at the selectivity of the antibody, but then we can't really disregard the selectivity of everything else within that molecule. We really need to understand exactly how to make it as selective as possible. And that may well move away from the antibody and move into different parts. And I think that's what we're really going to see over the next few years.
And I think that's the perplexing thing as we develop these molecules is that you have a selective antibody, you have a selective small molecule, but the way you construct it, the linker attachment point, the nature of the linker, even single atom changes can move us from a very selective agent with a decent therapeutic window to something that's non-efficacious, non-selective. I don't think we understand that yet, do we?
Well, I think to a certain extent we do, but not to the extent that we really need to.
Yeah.
I think with ADCs in particular, we've been so set on increasing the PD properties of the payload to be able to have a more selective approach. We're not actually considering the PK to the same level as what we are. So we really need to think about that in terms of the payload itself and whether we can, I think you mentioned it in one of the last talks, building a metabolic handle to be able to, once that payload's done its job, we clear it out as quickly as possible. And I think that's a really nice way of thinking about it, a really interesting concept.
But I think that's where that idea of a multidisciplinary approach, isn't it? Bringing in chemistry, bioscience, DMPK, ex vivo PK, understanding the subtle effects of these things in terms of that bispecific nature and therapeutic window, and how those subtle changes have a huge impact on the overall package property. You know, we're used to developing small molecules, and we're used to small changes having significant effects on PK and toxicity and overall therapeutic profile. It's an interesting concept to step back from that and look at it not just in that rule of five space, small molecule space, but in this huge conglomerate of an antibody, a linker, a small molecule payload, and how they all interplay together.
Yeah, absolutely. And I think what we're seeing, I mean, the rule of five is almost out the window these days with PROTACs and things like that. We're seeing such an evolution of small molecules. But once we bring an antibody into it as well, we should really start to think about, if we consider the rule of five as just a way in which we can sort of summarize all of the PK properties, PK in particular, then we can look at the antibody and we can look at the developability side of things. And that is a really key point. We're not saying we need to disregard everything that we say within the antibody or the ADC world and start to focus on the small molecule. It's a synergistic approach. It's looking at the antibody, the large molecule and the small molecule synergistically together, and how we can break those counterparts down to the antibody, the conjugation, the linker and the payload, and really understand how that works together and how that works as its own entity and not a small molecule, not a large molecule. It's a hybrid. It's a fusion of both of them together. So.
And I love your thoughts about degraders and other modalities. That feels like the topic for our next conversation.
I think so.